Hello everyone, welcome back to the session. So in today's class we are going to discuss about the another cell organelle, important cell organelle called the cytoskeleton. Okay, so in yesterday's class we have completed about the ribosomes. Okay, so in today's class, as a continuation of yesterday's class, we are going to see another organelle, another important organelle called the cytoskeleton. Okay, see what is cytoskeleton. So cytoskeleton is a complex network of protein filaments present in the cytoplasm of eukaryotic cells. Okay, again the cytoskeleton is present in the eukaryotic cells, and the cytoskeleton is nothing but a protein filaments, and this protein filaments will having a complex network, and that complex network of protein filaments we are calling them as a cytoskeleton. Okay, and that is present in the nucleus or the cytoplasm of the eukaryotic cells, and that will be the nucleus of the eukaryotic cells. Okay, so you should know that the cytoskeleton is a complex network of protein filaments that is present in the cytoplasm of eukaryotic cells. Okay, and what is the function of the cytoskeleton? So this is the one providing the structural support, shape, mechanical strength, and helps in cell movement, intracellular transport, and cell division. So all these are the functions performed by the cytoskeleton. The first thing will be the structural support. Okay, so the cytoskeleton is providing a support to the cell, and it is also providing shape to the cell and the mechanical strength to the cell, and helps in cell movement. Okay, the movement of cell has been done by the aided by this cytoskeleton, and the intracellular transport and the cell division. Okay, so for the intracellular transport and for cell motility, the cytoskeleton plays a crucial role for it. Okay, so and this cytoskeleton cell division is very important for the cytoskeleton. Okay, we are going to discuss about the cytoskeleton in the cell division very clearly. Okay, in cell cycle and cell division, the function of the cytoskeleton is immense. All right, now see, in prokaryotes, the cytoskeleton like element exists. See, what do we have studied here? It is present in the eukaryotic cell cytoplasm. In prokaryotes also, the cytoskeleton is present, but it will be a simpler structure. But in eukaryotic cells, the cytoskeleton is a complex network of proteins. Okay, complex network of proteins in case of eukaryotic cells. In prokaryotic cells, the cytoskeleton like elements are present, but it is very simple. Okay, and what is the cytoskeletal structures present in prokaryote means FITS Z, FTS Z, MREB. Okay, MEB and FTS, FITS Z. Okay, we can call them as like that. Okay, so these are all the cytoskeletal structures present in case of eukaryotes. Right now, so this is what a cytoskeleton. So in cytoskeleton we are having three important structures. One is actin, that is actin filaments or microfilaments. Okay, actin filament or microfilament, intermediate filament, and microtubule. All these three are the types of the cytoskeleton. Okay, so we are having actin filaments. So actin filaments will be look like this. Okay, so these are all the actin filaments, and the intermediate filaments will be look like this. Okay, so this is what the intermediate filaments, and the microtubules are look like this. Okay, so this is what a microtubules. Okay, so we are having three different types of cytoskeleton. One is microtubules, intermediate filaments, and actin filaments. So these are all the three different types of cytoskeleton. Okay, so now we are going to discuss about each and every one, their structure as well as their function. Okay, now overall, what is the function of the cytoskeleton? Okay, so the cytoskeleton maintains the cell shape and the polarity. Okay, so the cytoskeleton plays an important role in case of shape of the cell and the polarity. Okay, polarity in the sense plus or minus, towards the minus end, movement towards the plus end. So they are going to move from the minus end to plus end, from the plus end to the minus end. All these movement has been done with the help of this cytoskeleton, and we are going to discuss about that. Okay, and they are the one providing mechanical support, as we have mentioned before. So the cytoskeleton is providing mechanical support to the cell organelles. Okay, and cell motility. So for cell motility, the cytoskeleton is very important, and we have there is a locomotory organelle like cilia, flagella and pseudopodia. Okay, so these are all the locomotory organelles which is very important in case of cell motility. So what is the connection with the cilia, flagella and pseudopodia with respect to the cytoskeleton means you should know that the cytoskeleton, that is the which we are having three different types of cytoskeleton, and the one most important type will be the microtubules. And this microtubules will be present in cilia, flagella. Okay, so in the microtubules, that is the type of cytoskeleton which is present in cilia and flagella. Because of the movement of microtubules, the entire flagella or cilia tend to move. As a result, the entire organism will move. Okay, so that is why we are saying the cytoskeleton enables cell motility. Okay, now it facilitates intracellular transport of vesicles and organelles. Okay, so it facilitates intracellular transport of vesicles and organelles. So you should know the transport. Okay, so the vesicles and organelles have been transported intracellularly between the cells, inside the cells, that has been facilitated by this cytoskeleton. Okay, now this cytoskeleton is also involved in the cell division. Cell division. See, say for an example, in cell division, this mitotic spindle and contractile ring are very important for cell division. Okay. Now this mitotic spindle has been secreted by this microtubule, and this contractile ring will be the microfilaments. So the microfilaments and microtubules are very important in the cell division. Okay. Now it helps in anchoring the organelles. And the cytoskeleton helps in anchoring. Anchoring means support. Okay, first to support each and all the organelles, the cytoskeleton is very important. Okay, so far what we have studied: to maintain the cell shape. Okay, to maintain the to provide the mechanical support, shape and support, and cell motility, and to support anchorage. So these are all the most important functions of the cytoskeleton. Now the major components of the cytoskeleton. So as we have mentioned before, we are having three major types of this protein filaments. What are all those filaments? Microtubules, microfilaments and intermediate filaments. All these were the main components of the cytoskeleton. And see here, look at this picture. We are having microtubules. As the name suggests, these microtubules are in a tube-like structure. Okay, and it is made up of a protein which is called as a tubulin protein. Okay, see here, as it is made up of a tubulin protein, we can call them as this microtubules. Okay, and this tubulin, we are having an alpha tubulin and we are having beta tubulin. So that is why we are saying this microtubules is made up of alpha beta heterodimer. Okay, see here, this microtubules is made up of a tubulin protein. Okay, first thing. And this tubulin protein, again it is made up of alpha, there is an alpha tubulin is there and beta tubulin is also there. Okay, and this alpha tubulin and beta tubulin, we are calling them as this microtubulin is made up of alpha beta heterodimer. Okay, so this is very important, and the diameter of this microtubules will be 25 nanometer in diameter. Okay, now this actin filaments will be look like this. Okay, this is what an active filaments, and their now diameter will be the 7 nanometer in diameter, and there is an intermediate filaments, and this intermediate filaments will be, and the size will be 10 to 10 nanometer in diameter. And the question can be asked, well, we have to arrange all these protein filaments in an ascending order or it will be a descending order. The question make us either way. Okay, so you have to know that microtubules are very large and followed by intermediate filaments, followed by the actin filaments or the microfilaments. Okay, so this is based on the size, we can categorize these protein filaments accordingly. Okay, now comes to the microfilaments, that is actin filaments. We are going to discuss this microfilaments first, each and every filament. okay, and with their structure as well as their function. So this is what the structure of the microfilaments, and we have studied this microfilaments is also called as the actin filaments. And how actin is made up of, first you should know that, actin is a protein, okay, and this actin is made up of, actin is a polymer, okay, actin is a polymer and it is made up of two F-actin filaments, okay, it is made up of two F-actin. Two F-actin, we are calling them as an actin. Again, this F-actin is also a polymer. And how, what makes up the F-actin? So F-actin is also a polymer, and this F-actin is made up of many G-actin, okay. So you should know that actin is a protein and it is made up of two F-actin, and the F-actin is made up of many G-actin, and F stands for filamentous, G stands for globulin protein, okay. So you should know, so we are having F-actin, two F-actin together we are calling them as an actin, and F-actin is made up of many G-actins. Globulin is a monomer and F-actin is a polymer, okay. Now see here, what will be the diameter of this actin filament? Just now we have studied approximately 7 nanometer, and this microfilaments this microfilaments is very thin when compared to the microtubules and the intermediate filaments. This is very thin, that is approximately 7 nanometer, okay. And this structure, this microfilaments is made up of an actin protein, so that is why this microfilaments is also called as the actin filaments, okay. Now this actin protein is made, we are having two different forms of actin. One is a G-actin and the F-actin. G-actin is a monomer and F-actin is a polymer. And F-actin, F stands for filamentous, G stands for the globulin protein. See, G stands for globulin protein and this F stands for the filamentous protein. And see here, it is polar in nature. Polar in nature means one end is minus end and one other end is the positive end, okay, plus end, okay, and we are having plus end as well as the negative end. And see here, this plus end is called as the barbed end and the minus end is the pointed end, okay. See here, and this, what do we have studied? This actin microfilaments is made up of an actin filaments, and this actin microfilaments is made up of an actin protein, and this actin protein is made up of an F-actin, and this F-actin is made up of G-actin, okay. And you should know G-actin is the globular actin, which is a monomer which is making up the F-actin, that is a filamentous actin, which is a polymer, okay. So this is the important thing. And one more important thing will be this actin, they are having an ATP binding site, okay. ATP molecules, ATP binding site is also there, myosin binding site is also there in the actin, okay. Now, where this microfilaments is exactly located? This microfilaments is located in the located beneath the plasma membrane, that is in the cell cortex region. See, this is a cell. The outer region is called as a cortex and the deep inner region is called as a medulla. Always the cortex refers to the outer region and the medulla refers to the inner region. And this cytoskeleton, that is this the microfilaments will be present beneath the cell surface, that is the beneath the plasma membrane, okay. So more specifically in the muscle cells, because in the muscle contraction we need actin and myosin, the motor protein, the most important protein required for muscle contraction will be the actin filaments and the myosin, actin and the myosin, okay. And this actin and myosin we can call them as a microfilaments, okay. And in the microvilli of the intestinal epithelial cells, okay. So microvilli which is involving in absorption function, okay, and this microvilli which is present in the internal epithelial cells. So in the muscle cells and in the intestinal epithelial cells we can find these actin filaments. So the question can be asked where the location will be, okay, and it is beneath the plasma membrane in muscle cells and in the intestinal epithelial cells. So in these type of cells we can see the microfilaments rather than the microtubules and intermediate filaments. Okay, and we are having two effectin. Okay, two effectin we are calling them as an actin. And see here, so this is what an effectin, and group of G. So this is what a G actin. Many G actin we are calling them as the effectin, and two effectin we are calling them as the filamentous. As it is look like a filament, we are calling them as a filamentous actin. So if two effectin together, we are calling them as an actin filament. Okay. Now, what will be the function? So what do we have studied? First we have studied about the structure of the microfilaments, and we have studied about the location where it is located, and now we are going to see about the function of the microfilaments. Okay, so see here, very important, cell motility. This microfilaments is play an important role in the cell motility. And what is the type of movement facilitated by this microfilament means? That is amoeboid movement. Okay, now muscle contraction. So the muscle contraction, for this muscle contraction, actin and myosin are the two important proteins which is involving in the muscle contraction. Okay, so actin has the myosin binding site. So that is what, whenever the actin is binding with the myosin is pulling the actin, thus results in the muscle contraction. And the theory which is explaining the muscle contraction is the sliding filament theory. Okay, and the formation of cell cortex. For the formation of cell cortex, that is the outer region of the cell, the microfilaments are very important. And also, besides the motility and the muscle contraction, the formation of cell cortex, the cytokinesis, the formation of the contractile ring. This contractile ring is nothing but a microfilaments. Okay, the microfilaments forming the actin filaments, that is, it is also called as a microfilaments. It is making up a ring-like structure. We are calling them as the contractile ring. And you should know, wherever we can find the word actin, and this actin protein is involving in the contraction function. Okay, so that is why this actin filaments is present in the muscle cells and in the contractile ring. Okay, and also it plays an important role for the maintenance of cell shape. Now, not only for the cell motility but also for the cell shape, this microfilaments are very important. Now, the motor protein, that is the myosin. Okay, so as we have mentioned before, so this is an actin protein. So in the actin filament we are having a myosin binding site. Okay, so this is what the myosin, this is the head of the myosin, this is the tail of the myosin. So in the head of the myosin we can find the actin binding site as well as the ATP binding site. Okay, so see here, this is an actin and this is a myosin which is bind on the actin, and in the head of the actin we can also find the ATP binding site, so that in the muscle contraction process the myosin are in need of energy in the form of ATP. So that will be done by the ATP hydrolysis. Okay, so for this process, whenever the actin has been bind with the myosin is pulling the actin, and there it leads to the shortening of their filament, and thus results in the muscle contraction. And as I mentioned you before, this muscle contraction has been explained in which theory? Sliding filament theory. Okay, now see, that is what about the microfilaments. Now comes to the intermediate filaments. So the structure will be approximately 10 nanometer. And how this intermediate filament will look like? This is rope-like in nature and it is non-polar. So as we have mentioned before, in the microtubules we are having plus end and minus end, and in microfilaments also we are having plus end and minus end. And the intermediate filaments, we don't have any plus end and minus end. That is why we are calling them as the non-polar filaments. Okay, and this intermediate filaments are very stable and stronger than other filaments, and this can be expected in the question. So when compared to the microtubules, microfilaments, the intermediate filaments are very stable and very strong when compared to the other filaments. Okay. Now see here, this is what the microfilaments, microtubules and intermediate filaments. Just look at the picture. This microfilament is approximately 6 nanometer, 6 to 7 nanometer. We can take that as a 7 nanometer. Okay. And see here, this microtubule. This microtubule will look like a tube-like structure. It is look like a hollow. It is in hollow shape. Okay. And this hollow is made up of a tubulin protein. We are going to see about that. And this is an intermediate filament, and many protofilament together we are calling them as the intermediate filament. And you should know that all these microfilament, microtubule, intermediate filament, all of these filament are a polymer which is made up of some monomers. Okay, see, say for an example, this microfilament is made up of a tubulin protein, that which is a microfilament is made up of an actin, and actin is made up of many G actin. Okay, so G monomer. And in microtubulin it is made up of a tubulin protein, and the intermediate filament is again is made up of many monomer. We are going to see about that. So we have completed about the microfilaments and we have started the intermediate filaments. So as we have mentioned before, so when compared to all the filaments, these intermediate filaments are very stable and they are very strong when compared to other filaments. And what will be the composition and where this intermediate filaments is located, this we can expect in the examination. Okay, so in keratin, in the epithelial cells, we can find the intermediate filaments. Okay, so in vimentin, mesenchymal cells, we can find the intermediate filaments. And desmin, which is present in the muscle cells, we can find the IF, that is the intermediate filaments. And in neurofilaments, in neurons, and lamins, this is very important, this we can expect in the question for sure. Okay, so lamins, lamin protein together we can make up the nuclear lamina. So in all these areas, in all these cells, this intermediate filaments intermediate filaments are present in all these cells. Okay, now what will be the function of the intermediate filaments? Okay, see, this provides a tensile strength. So as I have mentioned you before, when compared to all the filaments, this intermediate filaments are very stable and it is very strong when compared to other filaments. Now you should know that automatically the function of the intermediate filaments to provide a strength. Okay, and to maintain the cell integrity. Okay, and to maintain the cell integrity means to make the cell stable, and to maintain the cell integrity, this intermediate filaments are very important. And it anchoring the organelles like nucleus. So anchor means support. It is supporting many organelles to maintain their shape and structure. Okay, so that is what the function of intermediate filaments with respect to the nucleus, maintaining the structure. And form the nuclear lamina. To this nuclear lamina, we can find below the surface of the nuclear lamina, we can find the intermediate filaments. And this nuclear lamina is made up of the lamin protein, and this intermediate filaments is the intermediate filaments is the protein filament, which is important in forming the nuclear lamina. Okay, now, what will be the special feature for this intermediate filament? See, whatever it is, for the microtubule, microfilaments, all these filaments, we need an energy. Okay, either it will be the ATP or GTP, but in the intermediate filaments, to perform their function, they do not require ATP or GTP for their assembly, which means they are an energy independent process. No motor proteins are associated here. Okay, so in microfilaments we have studied about the protein called the actin. In microtubules we have studied about the protein called the tubulin. But in intermediate filaments we didn't have any of the motor proteins associated with it. Okay, so this is very important point. Now, see, we have completed the microfilaments and the intermediate filaments. Now we are going to discuss about this microtubules. Okay, so microtubules, their structure is approximately 25 nanometer. Okay, 25 nanometer thickest, and it is made up of alpha tubulin and the beta tubulin. Okay, so this alpha tubulin and beta tubulin, as it is different, one is alpha and another one is beta. So that is why we can call this microtubule, we can call this as alpha beta heterodimer. See, hetero means different, dimer in the sense two. Okay, two monomer together we are calling them as a dimer. So here alpha tubulin is there, beta tubulin is there, that is why we are calling heterodimer. So this heterodimer, this dimer is making up the microtubules. Okay, and 13 protofilaments forms a hollow tube. See here, and we are having alpha tubulin and beta tubulin, and this alpha tubulin, beta tubulin. So this many alpha and beta tubulin, we are calling them as a protofilament, and this is one protofilament. This protofilament is made up of alpha and beta tubulin like this. Okay, alpha tubulin, beta, alpha, beta like this. And like in the same way, 13 protofilaments together we are calling them as the microtubules. Okay, and one end we are calling them as a positive end, and another one is called as a negative end. Minus end, plus end and minus end. And you should know that in plus end, the plus end is called as a growing end, and the minus end is called as a non-growing end. There will be no growth occurs in the minus end. Okay, and the plus end we can call them as the polymerization and depolymerization in the minus end. Okay, see here, 13 protofilaments forms a hollow tube. Okay, 13 protofilaments forms as a hollow tube, and the structure is called as a microtubules. Okay, one end is plus end, another one is minus end. And where this microtubule is originating, and this microtubules arise from the microtubule organizing center, that is MTOC. Okay, so we have studied about the nucleolus where the ribosome biogenesis is taking place, and the region nucleolar organizer region, from this region nucleolus has been formed. So that is like in the same way, they are having a microtubule organizing center. From that center microtubule has been originating. Okay, so in animal cells the origin is from the centrosome. From centrosome there is a microtubule organizing center will be there. From that center microtubule will be formed. Okay, and minus end anchored to the centrosome. And see here, and we are as I have mentioned it before, there is a plus end and the minus end, and from the centromere microtubule have been formed, and in the centromere, which end is anchoring, attaching to the centromere and the centrosome, that will be the minus end. Okay, now what will be the functions of this microtubule? See here, so formation of this mitotic spindle. Formation of the mitotic spindle. See, this is the chromosome, and this is the chromosome. The center point is called as a constriction. So in this constriction, centrosome is present. In the centrosome, we are having mitotic microtubule organizing center. From there, microtubule will be produced, and the microtubule, they are going to form the mitotic spindle, and this spindle is attaching the centrosome from both the ends. Okay, see, this is what a chromosome, and this is a chromosome. This is the mitotic spindle which is attaching the centrosome. Okay, so by attaching the spindle with the centrosome, the one chromosome will be separated into two. Okay, and the two chromatids present in one chromosome that has been separated with each other, and this is very important in case of cell division. In the cell division process, this segregation is very important, and that segregation has been mediated by this microtubules. How, in which structure? Because this microtubule is the one producing the spindle fibers, and that spindle fiber has been attaching with the centrosome from both the ends. But by attaching with the centrosome, the spindle fibers is separating the chromosomes. Okay, so the function of the mitotic spindle is very important in cell division. We are going to discuss about the cell cycle and the cell division. Okay, now the centrosome movement. Okay, see, the centrosome movement, for the movement of the chromosome, for the movement of chromosome, this microtubules plays an important role. And intracellular transport, for the transport process, microtubules are important. And for the formation of cilia and flagella, this is very important. So the microtubules are arranged in a cilia and flagella in the form of 9+2 arrangement. 9+2 array. See here, the microtubules will be occur like this. Okay, doublet microtubules. Okay, doublet microtubules. Okay, and in the center there is a singlet microtubules that like this. Okay, there is a in the periphery region it will be looking like this. Okay. Okay, in the peripheral region, two microtubules attached together, we are calling them as the doublet microtubules. In the center region, two microtubules that is not attaching, so we are calling them as the two singlet microtubules. So in the periphery region, 9 microtubules will be there, and in the center we are having two singlet microtubules will be there. Okay, 9 doublets plus 2 singlet microtubules. Okay, so that is what 9 plus 2 arrangement. So this structure is very important in case of cilia and flagella. Okay, so in cilia and flagella, because of the movement of this microtubules, so the because of the contraction of this microtubules, the flagella will move. As the flagella tend to move, the entire organisms will move. Okay, so this is why we are saying the cilia and flagella is considered as a locomotory organelle, right? Now, so this cytoskeleton, that is the microtubules, is also important for maintaining the cell shape. Okay, and we have studied the motor protein. See here we know that tubulin makes up the entire microtubules. Okay, and in the tubulin we are having two motor protein associated binding region, and the motor proteins is nothing but kinesin and dynein. Kinesin and dynein, this is very important. The motor proteins of microtubules is the kinesin and dynein. Okay, kinesin moves towards the positive end and the dynein moves towards the negative end. Okay, so this is very important. So the motor proteins, kinesin and dynein are the motor proteins which is present in the microtubules. This makes the entire microtubules. Okay, and you should know, we have studied this microtubules very important for the intracellular transport, and how all this transport is taking place because of the motor protein called the kinesin and dynein. Okay, so this is the membrane. Okay, and this is the center. From the center, that is negative, that is positive. Okay, so the kinesin always moves from the negative to the positive end. The kinesin always moves towards the plus end. Okay, this can be expected in a question. This is very important. Okay, and the dynein will always, see here, see, this is what a membrane, and this is the center region. This is the periphery region and this is the center. And in the center we are having a negative, and in the periphery we are having a positive. Okay, and we have studied this kinesin has been moved from the negative end to the positive end, because kinesin is a motor protein which is moves towards the positive end. Okay, and the dynein moves towards the negative end, from the positive to the negative end. Okay, so this is what kinesin and the dynein. Okay, so this is very important. This can be expected in the questions. Okay, okay, now cilia and the flagella. 1 minute. Okay, so now another important thing will be the cilia and flagella. And see here, the cilia and flagella, so this cilia and flagella is made by the microtubules. Microtubules make up the cilia and flagella. Just look at the picture. So this red color structure is considered as a basal body, and this basal body is also called as a kinetosome. Okay, see here, basal body, and this basal body is also called as a kinetosome, and from this basal body the flagella arises. Okay, and see here, whenever we cut the flagella like this, see this picture. Okay, see this figure 2, and in this picture we can see here as, see, this is one, this red color is 1, and this is 2, and this 3, 4, 5, 6, 7, 8, 9. As I have mentioned you, there is 9 peripheral doublets will be there in the periphery region, and in the center we are having 2 singlet microtubules which is not attaching with each other. So that is why I have mentioned you that it is 9 plus 2 arrangement, 9 plus 2 singlets. Okay, okay, and in the basal body, and in this basal body, see here, This basal body 9 triplet arrangement. This basal body has 9 plus 0 arrangement, and in the functions, the cilia, movement of fluids in the respiratory tract, in the flagella there is a locomotion. Okay, because of the movement or contraction of this microtubules, the entire flagella will move. If the flagella move, the entire organisms will move. Okay, so that is why we are saying the cilia and flagella is considered as a locomotory organelle. Very, very important, and you should know that. See here, and this in the microtubules in the periphery region, they have been present as a doublet. In the center region, they have been present as singlet. In the basal body, they are triplet. Okay, so this can be expected in the examination. Okay, and look at this picture. So see here, in this picture you can see clearly. So this is what a central microtubule. Okay, so this is the central microtubule, and this is the central region, and in the periphery region we are having two microtubules which is in a doublet. Okay, which is in double. So this is why we are calling them as a doublet micro. So this is what 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 doublet microtubules, 2 singlet microtubules. That is what 9 plus 2 arrangement. Okay. And here in the basal body, this is in the structure of 3 microtubules. See here, 9, 3 triplet. That is triplet microtubules is present in case of here. Okay. Now, in the cilia, so we are having another thing, that is, this is what an epithelium, ciliated epithelium. In the epithelium, so all these epithelial cells has been followed by the ciliary structures. Again, it is in the movement. Okay. Now, we are going to compare all the cytoskeletal elements now. So, these are all an important thing. Kindly make a note of it. Okay. See here, microfilaments, that is 7 nanometer. In intermediate filaments, that is 10 nanometer. Microtubules, that is 25 nanometer. Okay. So, when the question asked about based on the size, if they asked us to arrange in an ascending or descending order, you have to know that microtubules is very large, and followed by intermediate filaments, and after that microfilaments, and it also called as the actin filaments. Okay, and the protein which is making up the microfilaments is actin. The protein which is present in the intermediate filament is keratin and vimentin, and the protein which is present in the microtubule is tubulin. This can be expected. Okay, and the polarity is present in microfilaments and the microtubules, but not in the intermediate filaments. I have explained you this point. Okay, and the motor protein which is present, and the motor protein which is present in the microfilament is myosin, and the motor protein which is present in the microtubules is kinesin and dynein, and there is no motor protein present in the intermediate filaments. Okay. Now, the energy is required. So, this point also I have stressed when I explained you. The microfilaments need energy, microtubules need energy, and the intermediate filament doesn't need any energy. Okay, so this is very important differences. And see here, so as we have mentioned in the first 1 or 2 slides, we have mentioned that there is a cytoskeleton which is also present in the prokaryote, but it is not complex as much as in the eukaryotes. Okay, so the prokaryote, the cytoskeleton which is present in the prokaryote, which is very simple in nature. See here, FTS is it. Okay, so this is involving in the cell division. Again, it is tubulin like. We can call them as a modified form of microtubules. And MERB. Okay, so this is responsible for cell shape. Again, it is the actin like. So we can call them as a microfilaments. And CRES, curvature in bacteria. Now we can connect this with intermediate filaments. So these are all the cytoskeletal proteins which is present in the prokaryotes, which is simpler, not complex as much as in eukaryotes. Now see here, a very important slide. Okay, so this is very important slide. Why? Because so the inhibitors which is inhibiting the microtubules, microfilaments, intermediate filaments, all these are important. Okay, see colchicine and vinblastine. This is the one inhibiting microtubule polymerization. Polymerization means adding of the polymer, and that is the growing end, the plus end. Okay, so if it is inhibiting the microtubule polymerization, polymerization. So those inhibitors which is inhibiting the microtubule polymerization is colchicine and vinblastine. If it is inhibiting the actin polymerization, we are calling which inhibitor is inhibiting the actin polymerization is cytoskeleton B. And the defective dynin, if the dynin protein, see we have studied about this kinesin and dynin, these are all the two motor proteins in the microtubules, and this dynin is a protein which is moved towards the positive, moves towards the negative end. Kinesin which moves towards the positive end, and dynin is moves towards the negative end. Okay, if the dynin is not function properly, this dynin movement, that is towards the negative movement, primary ciliary dyskinesia. Okay, so this is what a syndrome. If the dynesin protein not function properly, that is, it is also called as the Cartaginer syndrome. Very, very important. This slide is very important. Now, so in the exam, definitely you have to remember about the diameters. That is 7, that is the microfilaments, 10 is intermediate filaments, 25 is microtubules. And motor protein direction is frequently asked. In case of kinesin, it is positive, it is moves towards the positive end. In case of dynin, it is moves towards the negative end. So this very important, and we have to know what is the 9+2 arrangement and what is the 9+0 arrangement. See, in the cilia and flagella, in the out periphery region we are having 9 doublet, and in the center region we are having singlet microtubules. This is what the 9+2 arrangement. And this basal body where the flagella arises, they are having 9 triplet plus no central microtubules. We are calling them as the 9+0 arrangement. Okay, the drug cytoskeleton matching question. See, for an example, the drug has been on one side and it is inhibiting on inhibitor is going to inhibit which cytoskeletal protein. So all these it can be asked in the mass of following questions. Okay, and these all are the one liner fact which is important for the CUET exam. See, the cytoskeleton is absent in the mature RBC. Okay, the mature RBC are absent in case of cytoskeleton, and the intermediate filaments are most stable when compared to the microtubules and the microfilaments. Okay, so when compared to other two filaments, so this IF is very, very most stable. And see here, this kinesin moves vesicles to the cell periphery. From the center to the periphery region, that will be done by the kinesin. From the periphery region to the center, that will be done by the microfilaments. Okay, the actin myosin interaction causes the muscle contraction. Okay, see, actin myosin interaction, how the actin is going to bind with the myosin, that cause the muscle contraction, as I have mentioned you before. Okay, so that's all about the complete information about the cytoskeleton. Now come to the question and answer. So which cytoskeletal element is directly involved in cytokinesis of animal cells? Okay, so they have so here they have explained asked the question with respect to the cytokinesis. Okay, so in cytokinesis, which cytoskeletal element is important? That will be none other than the microtubules, because the contractile ring that will be involved in the cell division. Cell division, cytokinesis means cytoplasmic division, which is nothing but a part of the cell division. So that definitely contractile ring of the actin filaments. Then the second question will be the polarity of the microtubules. The polarity of the microtubules is due to. So the polarity of the microtubules is nothing but the alpha tubulin and the beta tubulin, which is make up, that is the positive end and the negative end. And this is the plus end and this is the minus end, growing end and the non-growing end. Okay. So the polarity, what is the main reason for the polarity of this microtubules is nothing but it is due to the head to tail arrangement of the alpha beta diamonds. Okay. See here, which cytoskeletal component does not require any energy in the form of ATP and GTP? We have studied this exact statement, that is the intermediate filaments. Okay. A drug that specifically inhibits actin polymerization, which directly affect. See here, which drug is inhibiting the actin polymerization? That will be the Okay, so if the actin polymerization, if any drug, if it is inhibiting the actin polymerization, that is going to directly affect the mitotic spindle formation, no. Chromosome movement, no. And ciliary movement, no. That will be the cytokinesis, okay. Now see here, which motor protein moves towards the minus end of the microtubule. See here, which motor protein moves towards the minus end. From the plus end to the minus end, that will be the dynein protein. Okay, so that is exactly the option C. The 9+2 arrangement, just now we have studied, it is seen in the axoneme of the cilia and the flagella. And which intermediate protein forms a nuclear lamin? And well, just now we have studied lamin protein make up the nuclear lamina, and lamin is again an intermediate filament protein. Now, inhibition of this colchicine is immediately affect. So, the colchicine is immediately affecting the, okay, sorry, if inhibition of colchicine will immediately affect actin myosin interaction, cell cortex formation, spindle fiber formation. This is very important question. Actin myosin interaction, that will be done by the microfilaments. Okay. Cell cortex formation, that will be done by the intermediate cell cortex formation, again that will be done by the microfilaments. Spindle fiber formation, that will be done by the microtubules. Nuclear lamina integrity, that will be ensured by the intermediate filaments. And the colchicine drug is somewhat related to the polymerization of the microtubules. So definitely spindle fiber formation will get affected. So this is how you have to interpret what we have studied and what we have to choose, right? Which of the following correctly matches the cytoskeletal element and its function? Which is correctly matched? Intermediate filaments, that is intracellular transport, microtubules tensile strength, and microfilaments muscle contraction, neurofilaments chromosomal movement. Okay, so which is correctly matches? So that is none other than the microfilaments, which is involving in the muscle contraction with the help of actin. No. If a defect in dynein arms of cilia will primarily result in, if the dynein arm is not function properly, so dynein arm is present in the axoneme of the cilia, which is the one connecting one doublet to the another doublet. Okay, so if it is not connecting properly, what will the defect? That will cause the primary ciliary dyskinesia. Just now we have studied this thing. Okay, so that's all about the information about the cytoskeleton. In tomorrow's class, we're going to discuss about some other important things. Okay, thank you so much.